Yeah.
All right. Well, welcome everybody. I'm so glad that
can I, sorry, I'll first give the instructions and then I'll
Sure.
let you introduce. Just to give some background on what we're doing here today. Probably everyone might be a little bit confused. So last year, we, as the department, developed a Teaching Excellence in Entomology program. And what that means is that graduate students can use some of their time in grad school to work on their teaching skills. Mike is the first one to come through this program. This is his final step. So this is his master lecture. So if you're a faculty member attending, I sent you out Mike's syllabus and also the lecturer evaluation form. This evaluation form depends on faculty reviews for Mike to pass this portion of the teaching lecture. And for all of the students, specifically those that are attending the class, since you won't be doing the lunch today, what I would like you to do is you can either complete the lecturer evaluation form to give Mike some feedback, or you can just write out some brief comments if that's easier, but you need to send those over to me. It will be anonymous, just like regular class reviews are, but it's really, really important that Mike gets feedback from faculty and also from the students attending the lecture. So you can look at the syllabus, which I've posted in the chat. And if you'd like to do the official evaluation form, you can find that also in the chat. But if you want to just write out some brief comments or feedback, you can do that and send that to me after the lecture. So I'll hand it over to you, Dr. Moran, to introduce.
Great, thank you. And yes, welcome again, everyone. I'm glad you're able to join us on a Friday afternoon. Really, I want Mike to introduce himself a little bit because he's had a very interesting journey between his undergraduate days and now being in graduate school. He's a master's student in Dr. George Hamilton's lab, and he's now looking to do his PhD as well. And I first met Mike, gosh, has it been 2 years now? Oh my goodness, 2 years ago. I'm the project coordinator for something called the Arch Street Project. It was a historical cemetery in Philadelphia that was impacted by construction and resulted in a salvage archaeological project. And Mike took his own initiative to approach me to participate in that project and to do some entomological analysis on the human remains and the soil surrounding the human remains. So I had the pleasure of working with him for some time in my lab before COVID shut everything down. And Mike is just, a true educator. He has a wonderful ability to connect with the public. He has a real passion for his field. But again, I want to hand it over to him to talk a little bit more about his journey from A to B and why he has this passion for entomology. So Mike, I'll hand it over to you.
Thank you so much. Thank you so much for that introduction. I'm very humbled by that. Yeah, so I came into forensic entomology through my undergraduate program here at Rutgers, and working in forensic entomology, I eventually found my way to this even more niche subfield of archaeological entomology. And so that's what today's lecture is going to be on, is the study of insect remains found at archaeological grave sites. And we specifically call this funerary archaeoentomology. And I'm also going to take some time to talk about the Teaching Excellence in Entomology certificate program that this lecture was developed for.
And before Mike continues, I'm going to be monitoring the chat. So if you have any questions at any point, or if Mike throws out a question for you guys to answer, I'll be handing those answers or handing those questions over to Mike. Also, you can raise your hand as well, and that will pop you up in the gallery view, and he'll turn to you for your question. So take it away, Mike.
And we'll get all those questions answered, so feel free to ask them. So this lecture represents the final component of the Teaching Excellence in Entomology certificate program that was developed by the department to give graduate entomology students some more practical hands-on experience in undergraduate instruction. So through the course of this program, I've completed several teaching seminars through the Office of Instructional Design, or OID, the Center for Teaching Resources, or STAR, and I've done 3 teaching observations and 3 teaching lectures across several departments and programs in the university, including the undergraduate electrical engineering program and through the Department of Earth and Planetary Sciences. I submitted an original class syllabus for a quaternary and archaeological entomology class, and this would be the 10th lecture from that class. I originally wrote this class for in-person instruction, and throughout this lecture, I'm going to do my best to highlight some of the ways that I would make this material more conducive to an online format, like how I would structure my Canvas modules, the use of polls, and a few other strategies. So get ready to use that annotate feature in a little bit. This version of Lecture 10 is that we're doing is a little bit simplified because we haven't gone through some of that foundationary material together. And instead, I've replaced some of that depth of detail we would have gone over here with some extra background info that we would have already been quizzed on, and some of this information about the teaching certificate. So the original teaching philosophy document that I had submitted was written through the graduate writing program and edited by my program mentor. And so aspects of my teaching philosophy that I would like to highlight today are being mindful of the role of contextual factors, including remote learning and student accessibility. And to do this, for each week's module, I provide a concise overview which states the readings, the assignments, and the week's expectations, and importantly, the order that those tasks should be accomplished in. And I also want to highlight the nature of science by focusing on the interdisciplinary nature of archaeoentomology and the evolving limitations of the field. We'll start with an overview of today's talk, and I think this is particularly effective for distance learning, where pacing and screen fatigue can sometimes be a challenge. So we'll first touch on some key terms, and those would be this week's or this lecture's main vocabulary. Then we'll talk about some of that background information that I had mentioned. We'll cover it a little that we would have covered in a lot greater depth in previous lectures, but I think here it would be especially pertinent to making the overall topic more relevant and putting the entire class, what the entire class has to offer, into a little bit clearer context. And then we'll look at why study funerary archaeoentomology. Then we'll move on to some basic principles of sampling and analysis. Then we'll start winding it down with some notable examples, a summary of what we learned, and then we'll open it up for a Q&A. But of course, at any point, feel free to stop me and ask any questions. All right, let's dive in. So the assemblage is our first term, and that is the group of insects found in situ in our, or recovered from our site. A fossil insect, for our intents and purposes, is going to be any insect or arthropod remains recovered from your archaeological or geological site. Now, if you've ever even thought about a paleontology class before and the idea of calling something that's less than 3,000 years old a fossil, if that gives you really strong feelings, that is okay, that's fine. I encourage you to get involved and come debate it out at the next conference of the International Conference of Funerary Archaeoentomology. So NISP, or NISP, stands for the number of individual species present. And so this number is going to be the number of insect or arthropod species present in our assemblage. And this is related to the MNI, or the minimum number of individuals, meaning once you figure out that you have species X, the MNI is going to tell you how many of species X, or the minimum number of species X, you have in your assemblage. And this concept comes from traditional archaeology. And a good classic example of this would be the refuse pile at like a long-term encampment. So in our refuse pile, we might have the remains of one feline species, one canid species, one goat species, and one swine species. So 4 different species of animals, and our NSP is 4. And then to determine how many individual pigs we have butchered in that refuse pile, we're going to count non-replicable jaw bones. So for example, commonly what's used in mammals would be jaw bones or scapulas. So you would count the number of right scapulas you found in your refuse pile, and this is going to give you the minimum number of pigs you have at your site. And so calculating this for insects in your assemblage is going to be very, very helpful. However, it's a bit more complicated trying to figure out, an MNI of insect remains versus mammal remains. It's going to be much easier to figure out the potential mammal species you have in an assemblage versus the potential number of beetle species. But it is still very helpful. When evaluating MNI of an insect, we should remember that insects in an assemblage are rarely found whole. Instead, what you're going to find are fragments of insects. And what you're going to look for, those fragments you're going to look for, are usually the head, so the head capsule, the pronotum, a single elytron, a puparium, or in the case of the spiders, carapaces or cephalothorax. So when determining the MNI for each species, we will count only one of those non-replicable parts. So if you have a group of beetles, you're only going to be counting right elytron, you will only be counting pronotum. You will choose one of those parts. It is important to point out that parts like antennae, legs, very replicable body sclerites or cryptic body sclerites are not standard practice to use when calculating MNI.
I have a question.
Yes.
I'm a student in this class and I forgot what, can you go back to the previous slide? I forgot what cryptic sclerites mean. Can you re-explain that?
Yes, so that I could have used a more accessible term there. So when you look at the integument or the insect exoskeleton, you'll notice that it is not a single body plate, but there are many different body plates that are held together with softer interstitial tissue. And so if you were to look really close at an insect, you'll see that the body plates that are on the dorsum, or the part that is facing up, those sclerites tend to be larger. But if you were to look on the pleural or the side regions of an insect, those sclerites tend to be a lot more smaller and harder to place. They're harder to identify exactly what sclerite you're looking at. So when I say cryptic sclerites, what I mean is you would not use like smaller sclerites from the side region of the body, or like single ventral abdominal sclerites. Just are not characteristic enough. There are not enough diagnostic characteristics on them, and also it's harder to determine that there was only one. Like, for example, there's only one pronotum versus there being many different similar sclerites on the side, so that's actually what I mean. Was that an answer to your question?
Yep, thank you.
Great. Now I would move on to some background information, and in a typical lecture, I would just present this slide and quickly move through some of this background information that we would have already been quizzed on, just as a reminder, as a refresher. But I think that today I'm going to take some extra time to actually go through some of these concepts and why they are important in the broader sense of funerary archaeoentomology. So for those of us with a stronger entomology background, we'd be pretty familiar with our friend chitin. So for someone coming at archaeoentomology from a archaeology or geology background, they may not be as, they may not realize how vital of a role chitin plays in insect physiology, resulting in its ability to aid in the preservation of these insect remains. So chitin is a long-chain polysaccharide, functionally similar to keratin, and it is located in the outer layer of the insect integument. Chitin catalyzes scleroturization, or the hardening that allows for insect body plates or sclerites to be well preserved in depositional environments. Just like the role of chitin might be more apparent to an entomologist, thinking about depositional environments are usually the realms of archaeologists and geologists, but they're going to play, but depositional environments and the type of different depositional environments you could encounter are going to play a very large role in the analysis of archaeoentomology. And so these are just going to be a few things to keep in mind on a subject that's actually a pretty big topic. This is often its own class in geology or archaeology programs, and in this class, it would be its own lecture. So if you think about it, right, a forested environment that has a dynamic ecosystem in its leaf litter, right, where you have a lot of decomposition and breakdown going on, is going to be a lot less conducive to the preservation of insect remains than, for example, sediment settling at the bottom of a lake bed. And in a funerary sense, our different funerary sites are going to be their own unique kinds of depositional environments. And so beyond the natural depositing and shifting in a geologic context, archaeologists are also going to think about the man-made changes to the geologic profile. Think back to our example of the refuse pile at the encampment. So you have that refuse pile, which is human activity, and then you also might have things like shards of pottery too that might get mixed in there. And so that whole group of things, the animal bones and the shards of pottery, those are all examples of what is commonly called cultural deposition. So the type of deposition that is made by human activity. Other things that we really want to think about when we're considering depositional environments with regards to insect preservation are aerobic versus anaerobic environments, how much humidity is present, the freeze and thaw cycle, and the amount of mechanical pressure being exerted by the sediments physically compacting our remains. Dr. Hawkins, you had a question?
Yeah, why is anaerobic better for preservation of fossils?
It allows, so the anaerobic conditions allow for the yielding of the breakdown of the integument. So a very common is quaternary insect analysis from lake beds and from bogs. And so those environments tend to yield a high degree of well-preserved insect remains.
What is the process that enables that to happen exactly?
What is the process that enables the preservation to happen?
Right, of anaerobic specifically.
It doesn't allow for the bacterial growth and fungal growth that fungal growth especially, which is very damaging to insect remains.
Why?
Why is fungal growth damaging?
Mm-hmm. No, why doesn't it allow those conditions? What are the conditions necessary?
For you need the air and nutrients. Does that answer your question?
Yeah.
Just like we learned, most of your specimens are going to be recovered in fragments. Beetle fragments are some of the hardiest remains. They are some of the most highly sclerotized remains, which allow them to be the most abundant taxa, typically represented in a historical or ancient assemblage. We won't be able to identify all of the remains down to the species level without an intact whole body, but it will be possible at times. And even a family level ID is going to be able to give you a lot of information about your site. The way that we're going to be identifying these beetle fragments is through the microsculpturing of the exoskeleton and other structural characteristics, like the shape of the elytral apex, or the arrangement of where the setae are or setae would be, ridgy striations, and the shape of the shoulder tooth. In 1948, a Swedish scientist named Lindroth was the first to document and then validate the use of microsculpture patterns to identify beetle specimens. On the right, we can see a scanning electron microscope image of a beetle elytron, and that stippled round kind of texture you're seeing is what is referred to as mesh. And different types of mesh on the elytra can be diagnostic and able to help you identify them. So certain techniques are going to allow you to view these characters without the aid of a scanning electron microscope. For example, turning the brightness down on your light source, changing the angle so that the angle of the light is coming more perpendicularly, and then diffusing the light source with parafilm over the light source could aid you in actually seeing these characters under a microscope if you do not have a scanning electron microscope. Although obviously a scanning electron microscope will bring out these details really well. The puparium of flies is a lot more difficult to morphologically identify than beetle fragments. And that being said, the pupal casings of true flies are the most sclerotized life stage of a true fly, meaning that the puparium is the most likely evidence to survive in an archaeological deposit of flies. Of flies. And at times you will be able to determine some morphological characteristics. So for example, on the left, you see an example of the life cycle of a phorid fly. So phorids are tiny true flies, and at least one common name that some phorids have is coffin flies. And that's because they have the ability to burrow down to or access carrion that is usually concealed and inaccessible to larger flies like blowflies, flesh flies, and musket flies. And so if you notice the dimension on the pupae image, you can see that these pupae are very tiny. And so that's going to be one of the morphological characteristics of this group of flies, is how much smaller their pupae are compared to other true flies that would be found in a mortuary site. All right, now that we've gone through all of that background information, let's actually get to some of the content from today. Now, what is a funerary site exactly? A funerary site is, plainly put, any location where one or more people have been interred after death. And throughout human history, we see a really wide range, a lot of diversity in the different funerary sites that human beings have used. So for example, you have really elaborate stone tombs like that were used in the Valley of the Kings in Egypt, or the De Medici burials in Renaissance Italy. The ritual use of open pits was employed by the ancient Zoroaster people in what is today Iran, and the Moche people in the high plateaus of what is today Peru. Cave burials have been documented all throughout the world, and you can see an example of two funerary bundles that were examined for insect evidence that were recovered from Mexico. And of course, what we think of as the traditional graveyard that has been the standard across North America since colonization also represents a very common type of funerary site. What is important to keep in mind with regards to insect ecology is that beyond the different geographical regions, different funerary sites are going to create different microclimates, which may either attract or repel different groups of insects. So for example, those funerary pits I mentioned earlier, in the case of the Moche of South America, they deliberately left their funerary pits open to flies and maggots, versus something like a cave burial might be more accessible to collembolans and phorid flies than it would be to larger flies in most beetle species.
Mike, there's a question from Sandy. She wants to know, by open pits, do you mean fire or refuse pits?
No, I mean, that's a very good question, Sandy. Thank you so much. I know you have a really strong archaeology background, so I know exactly like where you're coming from. So actually, so the Zoroaster people, I believe they had a special name for it. I believe it was the Tower of Tears or something. They believed that it would contaminate the ground to bury people. So they would actually form these open pits where vultures were able to come and deflesh the bones. And it was their belief that vultures were sacred. And they actually had a very similar view of vultures as the Moche people in South America, who viewed flies as sacred. And we'll get into that a little bit. So these pits that I'm referring to were areas that were open where fresh human remains were placed in there and just allowed to decompose either through birds or through maggots. That was a really good question. Thank you for bringing that up. All right, so I would normally use a poll right now. If possible, I would like everyone to type your favorite season in the chat box. This is something that I was going to set up beforehand, but there's settings in the Zoom, so I wasn't able to set up a poll. And the reason I'm doing this poll is because I like to use at least two simple polls throughout an online lecture as a soft measure of engagement and attendance. So by tracking who is consistently answering polls throughout the semester, I'm able to gauge, who is engaged and who is consistently coming to the class. So I'm actually seeing a really big mixed bag. Usually fall, which is my favorite season, wins, but it looks like we're skewing a little bit towards spring this time. And I love the different answers. And why that's important is because just like you have a seasonal preference, most insect species do as well. And so these seasonal preferences of insects within your assemblage are going to be analyzed in aggregate to determine seasonality. Or when I say seasonality, what I mean is the season that our deposit most likely occurred. Then beyond that, Elias in his 1994 book pointed out that the ecological niches occupied by most insects has remained relatively unchanged for the last 2 million years. So if you are able to evaluate what niches are represented in your assemblage, you are then able to make some determinations about the type of climate pattern you're looking at in your site. And now finally, pest presence. So it's relatively common for pests to be present in an archaeological assemblage. And then by evaluating the number of different pest species and the abundance of those species, so your MNI and NISP, you might be able to draw some conclusions about the severity of the pest infestation. And why? Why does that matter? Why does it matter what season, was occurring? Why does it matter, what the climate was like? Why does it matter what pests these people? I will tell you why it matters. It matters because of, the climate conditions you live in, the pests that you live with and battle with every day. Those are all factors that impact your health and well-being. And they are going to be factors that impact the health and well-being of the people that our site is studying. So consider the health and well-being of, consider the health, I'm sorry. So considering the health and well-being is going to be a major question of most archaeological investigations. And chances are the project that you're assisting with, the site, the site director is going to be considering the health and well-being of your site's people, because generally in archaeology, that is considered a contemporary issue in archaeology, determining what factors affected the health and well-being of your people. So interpretation of the insect evidence, your interpretation of the insect evidence is going to be combined with the interpretation of other physical evidence. So you, as the entomologist, are going to interpret the facts as best you see them. And then that's going to be correlated with things like pollen analysis, bone analysis, pottery and material culture analysis. And then the sum total of these things are going to allow a qualified person to then draw conclusions about the health and well-being of your site's people. So in this regard, archaeoentomology can provide a really significant piece to the puzzle that is an investigation of the past. Let's move on to some sampling. So I actually, let's break out your annotate feature. So what I would like you to do, all right, in there, let me get a little ahead of myself. In their textbook on forensic entomology, Bird and Allen outlined that the sampling of in situ historical remains is going to follow the same format as sampling for a regular contemporary modern forensic entomology case. This is something that we would have covered in module 8. I'm not giving you any preparation. I'm just throwing you into this. So if you would like to take out your annotate feature, you can either use the stamp or the draw feature. And I want you to kind of mark up maybe where you would, so maybe I would take a sample from here. So if anybody wants to participate, that would be great. Just take a few minutes and see if you could intuit where in this area you would take some soil samples from.
And Mike, you just might need to explain to folks how to access their annotation.
Oh, I'm sorry. So if you go to the top of your screen, there's 3 dots that say more, and click on more. And then if you scroll down, annotate is one of those options.
Are you on a Mac or are you on a PC?
Oh, I'm sorry. The other option, right. So sometimes that screen bar, your option bar, just has the annotate feature directly on it. So if you click that annotate feature, another option box will show up with draw, stamp. And so once you access those, you'll be able to mark up the page. All right, it looks like we are. All right, it looks like we are getting some pretty good responses. All right, looks like we're gonna have all the ones that we have today. Thank you so much for everybody who participated in this. So Bird and Allen state that historical sampling will follow the same format as as you would a modern forensic entomology case, except instead of collecting live insects, what you're going to be doing is taking soil samples. So a lot of you had the right idea. Exactly. I loved how you, I saw soil samples being taken from the head region, which is exactly where I would take soil samples from, 3 regions around the head, maybe the front, the back, the apex. I saw some examples of chest cavity, which I would take samples from the chest cavity, the pelvic region, as well as like the legs, hands, feet, and the margin. So that big oval I have there represents the margins of the burial. So for example, if you had something that was like a wooden coffin that has since decomposed and rotted away, you might actually be able to determine the delineations of that coffin by where the fly pupae are located. And so you'd want to sample that marginal region for fly pupae, because as fly maggots finish feeding and become 3rd instar maggots, they leave the food source and try to travel away to pupate. In this situation, they're going to be blocked by the actual coffin. They're going to meet that barrier, and that's where they're going to stop. They might crawl up the coffin, right, and pupate on the sides, but regardless, they're going to be stopped, halted by the barrier of that coffin. So sampling where you believe the margins are of the burial pit is going to be helpful for potentially locating fly pupae. If possible, you'd also want to sample, also want to examine the actual skull cavity for insect remains and the actual bones themselves for any damages or changes that could have occurred, like that has been documented with the damages that human bone receives when it is fed on by either subterranean termites or different types of beetles. And so the changes that bone goes through as insect damage is inflicted on it is usually termed insect taphonomy. Another thing you would want to examine are any funerary objects that are in direct proximity to the remains. So for example, chemical imprints of beetles have been found on belt buckles, swords, and other metal funerary objects. On the right, we see an example of some fly pupae that were imprinted on a piece of pottery through a chemical reaction. And another example of this that I'm going to bring up a little bit later was an example of an entire beetle found in the biscuit in a biscuit recovered from an Egyptian tomb. And so the changes caused to historical bone by insects and insect taphonomy and the examination of funerary objects from an archaeological entomology perspective are subjects that would be covered in another lecture. So sampling, some more sampling. The modern sampling of the site should be undertaken if possible. So trapping methods for modern insects include carry-on traps, pitfall traps, sweep netting, and beat sampling. You should also, if you can, you should also conduct a literature review of that region's insect literature, specifically focusing on pest management literature. So let's talk about processing your samples. In a funerary site, the samples are going to be very small. And so they are going to be sifted by hand under progressively smaller sieves. This is not the sieve or the technique I would use. However, in class, we would be handling most of these objects and actually doing some of this work together. In the distance classroom, we can't do that. So I've chosen to use some GIFs as a means of allowing these objects to be seen in a 3-dimensional space, but also as kind of like a soft attention grabber. Each catch is then going to be decanted into a petri dish and then examined under a dissecting microscope. And you can use a paintbrush with 80% ethanol. So what you would dip the paintbrush in the ethanol to grab the specimens off of the petri dish plate. Curating and storing. So depending upon your the condition, size, and the level of degradation of your insect samples, your insect fragments, there are two primary ways that you can curate and store them. In 80% ethanol or point mounted with some water soluble glue are the two most consistent methods for curation. One thing to keep in mind is if your samples, if it's possible to do any genetic testing on your samples, you should consult with your molecular scientist to determine what the best plan, to determine a best plan for curating and storing your samples so that genetic material would still be available and as undamaged as possible. Databases provide a computational approach to genetic climate to creating a genetic climate profile, generating a climate profile using the insects. And every entry into the databases strengthens them by adding more data. And this is where your MNI and your NISP will really come in handy in helping to determine the range and abundance of taxa will then help the computer program create a more accurate profile. Examples of databases that are helpful for this type of work are the Bugs CEP package developed by NOAA and Umeå University in Umeå, Sweden, and the Neotoma Paleoecological Database developed by the National Science Foundations and our international partners. As time goes, advances in analysis of ancient DNA or aDNA allow us to perform molecular identifications on some samples. And at this time, for various reasons, molecular dating of samples is not very feasible. In addition to using databases to aid in generating profiles of your assemblage, using your modern sampling is going to be able to tell you a lot about your site. For example, if your modern catches are consistent with your historical assemblage, then that may be indicative that there has been a relatively stable ecology at your site over a long period of time. However, if your catches are vastly different, this might suggest a more dynamic ecosystem. That being said, seasonality, our good friend seasonality, has to be controlled for, and incidentals. So in 1976, Kenward developed a method for identifying and removing incidental insects from your assemblage. What do I mean by incidentals? So not every insect you find in your archaeological assemblage is going to be representative of that site as a whole. If you think about it, you know, most of our insects fly, even if they don't mean to. Most of our insects fly, so even if they're going from point A to point B, they may still stop in the middle and then become incorporated into our assemblage. And what Kenward did was figure out a way to control for and remove those entries so that your analysis is actually representative of your site and is not being skewed by anomalous specimens that are in your assemblage. So one other thing that we need to keep in mind is that even though beetles and flies are the stars of our show, a lot of different arthropods can be found in an assemblage, and especially in a funerary assemblage, spiders, myriapods, mites, and snails can really add to an assemblage's richness. So in modern forensic cases, what we're going to be looking for is a postmortem interval or a time of colonization. We cannot determine those things from our historical assemblage. What we can do is determine or try to determine our old friend seasonality. So instead of determining the actual date when our assemblage was formed, what we're going to be doing is looking for the most likely time of the year that our assemblage was deposited. A classic example of this is the presence of certain fly remains in the bison skulls found at an indigenous burial site in South Dakota called the Akira burial site. And so the specific species of sarcophagids, of flesh flies that were found in the skull were indicative of a warmer spring climate. I mentioned before a beetley biscuit, so a whole beetle that was found in a biscuit. And that was actually recovered from an investigation of funerary objects that were located with an Egyptian royalty. So these items were now kept in repository in the Egyptian Museum at Turin. And so in 2003, an investigation was conducted of some of these objects. And one of the more interesting findings was this beetle that was embedded into a biscuit, ancient biscuit, like a 2,000-year-old biscuit. Now, one thing that archaeoentomologists consistently report is that ancient insect remains have a quality and character difference in them versus modern remains. Now, that's not very, can't really quantify that. You can't really quantify character in the same way that you would be able to do an investigation. So, for example, this researcher, Pangia Takapulu, what she did is she conducted a very thorough pest investigation of the repository and the museum at whole to confirm that no modern infestations of these beetles, these biscuit beetles, existed. She also took this specimen and compared it to modern biscuit beetles, so other examples from collections in museums, as well as freshly caught specimens. And that's how this specimen was validated as authentically ancient. I also mentioned the pre-Columbian Moche civilization earlier, and they were very distinct for their use of ritual open pit burials. And we don't know a lot about their culture, but we do know that flies and their maggot larvae played a huge role in the Moche's cosmology, or their spiritual view of the wider world. In their pottery and other art, we see images of people dancing with flies. And here we can see on the left a copy of some Moche art. And it is believed that the Moche believed that flies carried people off to the land of the dead. So once the maggot consumed you and turned into a fly, it was that fly that would carry you off to the afterworld. And we can see what is interpreted over here on the left as like a fly laying in wait, while these individuals, possibly skeletons, perform a sort of like dance macabre. An extensive analysis of the ancient insect remains associated with the Moche burials was completed by Uche in 2014. And one thing that was really interesting that was confirmed through this investigation was that the species Cochleomya macellaria was definitely present in this location in South America prior to European contact. So a quick recap. Today we learned that different groups of insects will preserve better than others, with beetles and fly pupae being our most abundant taxa represented in our assemblages, but lots of different other arthropods can add a rich value by adding a depth of diversity to our assemblage. Sampling of human remains can follow the standard forensic entomology principles, where soil samples are taken from representative regions of the body, including the edges of the funerary receptacle. Extensive companion sampling of the site's modern insect fauna should be done when possible. Ancient soil samples are processed through hand through progressively smaller sieves before being sorted under a microscope. Proper curation and storage is important for the longevity of your samples. And finally, databases and ancient DNA allow modern archaeological entomologists to create habitat preferences and identification profiles of their samples while updating the data in real time for the benefit of future researchers. Before I open up everything to a question and answer period, I do want to take a few minutes. Thank you everybody for joining me, and I'm going to thank all of the amazing people who helped and guided me through this journey. First, my program my teaching program mentor, Professor Kimberly Moran, the program lead of the Forensic Science Master's Program at Rutgers Camden, who has allowed me to do classroom observations and reviewed my teaching philosophy and my syllabus, and just has put so much work into me and getting this, bringing this to fruition. And I'm extremely grateful. Dr. Lauren Adamo, Dr. Mark Robson, and Dr. George Hamilton all allowed me to deliver required lectures from their syllabi in 2020 as a guest teacher. Dr. Madhav Kalfle was my instructor through the graduate writing program. Professor Tatiana Rodriguez and Annalise Saccioli allowed me to do classroom observations, and you saw some of the strategies I learned from them today. The amazing instructors over at OID and STAR, who hosted lots of classes that shaped my view of instructional delivery. And specifically, I would like to thank Ruth Ronan and Marcy Ansberger there. Of course, thank you to Dr. Wang and Francine for hosting me today. And of course, last but certainly, most definitely not least, thank you so much, Dr. Chloe Hawkins, who put together, organized this entire program that I found extremely educational. And I think that, you know, anybody in the graduate program should definitely at least, you know, try to complete this, because I mean, there was a lot of work, and I feel that at the end of this, I am a completely different, my view on instruction and my delivery methods are a world different than when I started. So thank you everyone for joining me. If you have any questions, I would love to open it up for questions now.
And you can ask questions on the content of today's lecture. I did put the syllabus, so if you have questions about the syllabus or the learning goals or any of the teaching pedagogies that may go along with this, you can ask those too.
I think Sandy has a question.
Great. Sorry. All right. Sandy had a question.
Sorry, I don't know why my audio always does that. But hello. Yeah, I love this presentation. It was really cool to see someone else in archaeology doing something in this department. I hope you don't mind if I kidnap you for the Rutgers Archaeological Society, because I've been trying to get them into bugs for like the longest time. Yeah, I did have a question. So yeah. For forensic anthropology, we deal with obviously like human assemblages, right? And we determine MNI by measuring bone length and determining how many individuals there are by like those proportions, right? But for insects, this is obviously like, you know, much more complicated. And you did say that you often get fragments, but like how exactly are you matching fragments to an individual? Like, are you measuring them by like body proportions, or like what is the process in the lab for doing MNI?
That's a really good question. So you would assemble all of your fragments and then start trying to identify, like first you would try to loosely sort of identify, try to place them into families if you can. And then beyond that, that's when you start determining, okay, like these, then you would actually take you could actually take a caliper. A wheeled one definitely works better. And you would actually start to measure these fragments. And so when it comes to, so the measurements, we're actually looking for measurements to see if these fragments are consistent with each other, rather than like trying to put together a whole body. You'll never be able to even put together a whole insect body, or even part of one, or even like 30% of one, right? Like, I mean, really all you're going to have are just like those head capsules, pronotum, elytra. So you aren't going to be measuring them for trying to determine like an individual, but you actually would use measurements to kind of confirm that, like, can confirm an MNI. So if you look and you have like one pronota that is like the proportions are like a lot different than the other ones, and you can go back and confirm that, you know, statistically 4 out of my 5 measure 2.3 millimeters, and then this one is different, you know, that might be an indicative that you might actually be dealing with a different species. So doing MNI on insect remains, definitely a lot more complicated, way more complicated. And it's not as, because this is such a small field, you know, you only have like a handful of people who are doing this. And so what we really need is a lot more people doing MNI with insect remains, so we can really standardize these practices. I would also say that too, that right now, the practices aren't like extremely standardized across the board. And the main reason that is because, I mean, if you were to take all of the funerary archaeoentomology people, you could like put them in a small room together, like a really small room. And so there's just not enough people right now to be winning those grants and doing that kind of work. But thank you very much. That is a really, really good question. And if you have any more questions, please feel free to ask.
Yeah, thank you.
So Mike, I have a question. Are you able to do all the lecture online? Because I thought that there may be some hands-on materials that you have to show people, right?
So I've been really thinking about that, and my my inclination is that it is possible to do online if with some sort of like a lab fee. So what I would actually have to do is assemble kits. So I actually do have like some like mock soil samples where like I put actual fly pupae in them, and you have to like sieve them out. And so like I have designed like kits that you would be able to send to people or have people pick up so that they could do some of this work at home. But yeah, it definitely would be a detriment to do this in a distance format, because, you know, just there's so many hands-on things. You would not be able to get the full benefit of this through online. But I have been trying to think about ways to at least mitigate that if this were to be handled in an online format.
Okay, thanks.
Thank you.
Mike, I had a question.
Yes.
So at the beginning, you were talking about how you want this class to be both for like entomology people and for our archaeology people, right?
Mm-hmm.
So I was looking at your syllabus. Do you think you would benefit from having a like insect biology or morphology lecture, so that when you're talking about all these different concepts, that would be covered, especially if you're bringing archaeology?
I must have been not clear about that. But they're in the first 3 modules, we would be going over like insect morphology and insect biology, just not very in depth, because again, it's just the integument, it's just the exoskeleton that we're mainly going to be dealing with. But I would set aside a certain day for complete day for beetle morphology, a complete day for fly and other species morphology. But then I don't think we would necessarily be going into physiology as much, because, you know, you wouldn't be able to see any of that in your assemblage. What you're just looking at are the integument and the exoskeletal remains. But thank you for pointing that out, because that is very important. So like my idea for the class is that in the first couple of modules, I do believe it's within like 2 to 4 modules, 2 to 4. Within those, there would be lessons on insect structure that is specifically tailored to looking at and identifying these structures in an assemblage. Thank you very much for that.
Does anyone else have a question? I have a question if no one else does, but I wanted to open it to everyone first.
Yeah, I have a question actually. Like, what kind of a role do ectoparasites play at these sites? Because I think I had read some reports about, you know, the travel of fleas from the old world to new world through the burying bridges and things like that, and the way that they kind of pieced that back together was by finding them at grave sites along the way. Yep. So are ectoparasites commonly recovered at these kinds of things?
Oh, yes. Fleas have been recovered. So there was work, which was actually a big deal because they're soft-bodied. So the fact that they were able to recover some flea remains was like a pretty big deal in the archaeoentomological literature. And so looking at parasites is definitely a huge role in this, because that speaks directly to health and well-being. So that is something that, if you're looking at an assemblage, you're going to pay a significant attention to that. For example, I'm currently working on a project in Maryland. And so it was 2 tombs that were buried in 1680. And so the one, the male individual has a very large abundance of mites on him. And so one of the things that we're trying to do is we're trying to go back and try to see if we can figure out, like, are these mites related to his business dealings? Like, was he out, interacting with other people? What kind of business was he in? And so we're really trying to look at that. But ectoparasites are a huge deal. I know there was a paper published from Egypt. I believe that was Pangia Takapoulou and Uche, who did an analysis on a mummified dog, and they spent a significant portion discussing the fleas and the different ectoparasites on that dog, and then relating them to the modern populations of parasites in that region. So that is a huge part of this type of analysis.
Cool.
Thank you.
Okay, I have one question, and then I think we can end it. My question is, what are the specific learning goals for this class, and how do they align with an entomology department's learning goals?
Some of the specific learning goals for this class would be to learn how to actually function as somebody in doing entomological analysis at a funerary site. So this is a very interdisciplinary class. Obviously, after one class, someone isn't going to, I don't think someone would be, directing an insect analysis at a site, but this definitely gets people involved in insect analysis of insect morphology, which is definitely one of the learning outcomes of a entomology department. Familiar with different trapping and sampling methods, and familiar with broad concepts of insect ecology and how insect ecology can be extrapolated to an interdisciplinary study. So this whole field of archaeoentomology is extremely interdisciplinary. And so a lot of times as insect scientists, we're kind of like, we're forgotten about sometimes. And so one of the goals of this class is to draw in students from other departments, from ecology, from archaeology, from geology, and to give them some foundation in insect science, but then to have them leave and remember that we exist. And so in the future, hopefully with more people being exposed to this, it's going to broaden the field of archaeological entomology and interdisciplinary science.
Great, thank you, Mike. Before we end, I would like to remind everyone that can, please complete the evaluation form. It's especially important for faculty in our department to complete these forms because Mike needs a kind of pass from at least 80% of the faculty in attendance today in order to achieve his certificate. So thanks again. And if anyone is a graduate student interested in this program, please let me know. I can add you to the Canvas site. You can spend your whole degree doing it. There's no time limit on when you can start and end.
I highly recommend everybody participate. If you have no other questions.
Okay, I guess we can stop here. Thank you, Mike. That's a great presentation.
Thank you. Thank you for having me.
All right. Thank you. We can leave then.